Hello, everyone. I'm Dr. Jack West, medical oncologist and founder and CEO of the Global Resource for Advancing Cancer Education, or GRACE, a nonprofit dedicated to free, timely information about cancer for patients and caregivers provided directly by the experts. Today, we're in Santa Monica at the Targeted Therapies in Lung Cancer meeting, sponsored by the International Association for the Study of Lung Cancer. We're actually here for this program in partnership with Longevity Foundation, who works on research, advocacy, education, and support. We're here today to bring you what I hope and expect will be a terrific webinar program on molecular testing in advancing non-small cell lung cancer, who to test and what to test for. I'm delighted to be joined today by 4 leaders in the field who will bring different perspectives. We'll see where we converge and we'll see where we actually have some differences in our opinions. Those 4 people are Dr. Charlie Rudin from the Sidney Kimmel Cancer Center at Johns Hopkins University in Baltimore, Maryland. Thanks for coming.
Absolutely.
Dr. Alice Shaw, assistant professor and thoracic oncologist at Massachusetts General Hospital and Harvard Medical School in Boston, Massachusetts. Thank you.
Thank you.
Dr. David Spiegel, thoracic oncologist and director of clinical research and lung cancer program at Sarah Cannon Cancer Center in Nashville, Tennessee. And Dr. Glenn Goss, who's the chair of the Lung Cancer Committee of the National Cancer Institute in Canada and professor at the University of Ottawa in Ontario, Canada. Thanks. Just to start with, one issue that comes up is the terminology of the various tests. We have immunohistochemistry, we have FISH analysis, and we have gene mutations. Charlie, can you give a little introduction of what are the differences when we're talking about these different tests?
Sure. So usually when we're talking about mutations, we're talking about alterations on a very small level in the DNA, often single substitutions of the coding sequence of genes. So DNA is comprised of long molecules, and a segment of that will be a gene, and there can be changes within that gene that essentially represent mutations. We're able to detect those mutations now by sequencing directly the DNA, which gives us insight into changes that are driving mutations for particular tumors. The other type of analysis that we frequently do to look at alterations in the DNA is the one that you referred to as FISH, which is fluorescence in situ hybridization. This is a technique where we use fluorescent probes that bind to the DNA that can look for rearrangements of the DNA. That is a more global mutation of the chromosome, where the chromosome actually rearranges to form new genes that have driving properties for cancer.
You were also part of the Lung Cancer Mutation Consortium, and I think it would be very helpful if you could highlight some of what that was, both the specific study and then what the implications were of the work, because I think that it's really reverberated through the field beyond 14 centers and 1,000 patients. So I'm going to turn it to Dr. Rudin to speak on a bit of introduction to the Lung Cancer Mutation Consortium project.
Well, thanks, Dr. West. I'd like to thank both Grace and Longevity for putting together this discussion today. I think it's quite a nice topic and clearly a very timely one. Just to briefly review the Lung Cancer Mutation Consortium, this is really a collaboration led by Paul Bunn at the University of Colorado, bringing together cancer centers around the country to work together looking at these mutations that we think are driving lung cancer. So the objectives of this consortium at the outset, this was really put together in 2009, was to characterize 1,000 tumor specimens from patients with lung cancer of a particular subtype, lung adenocarcinoma, which is the most common type of lung cancer in this country, for a number of driver mutations. These are mutations that we think are not only present in the tumor, but very important in deriving cancer. A list of the 10 changes are shown here. Most of them are mutations. A couple of them are alterations in the structure of the DNA. The last 2 there, EML4 and MET amplification. The goal here was both to analyze 1,000 tumor specimens to characterize the frequency of these mutations, but also to use that information for patient care, that is to recommend therapy or clinical trials based on the specific mutation identified. This is a snapshot of the centers that participated in this consortium. As you can see, they really span the country and many of the major medical centers. And the organization was really put together by Paul Bunn, and I think he deserves a lot of credit actually for being able to pull this together very quickly. This brought together many of the institutions that have large NCI grants, National Cancer Institute grants, to support lung cancer research, as well as the NCI itself. At the outset, I think the goal was seen as very ambitious to sequence 1,000 patients and characterize them, and I think there was some concern about whether we'd really be able to do this. This is sort of how the data from the Lung Cancer Mutation Consortium works. Lung cancer patients are enrolled in the study. They have to be of otherwise reasonably good health and have available tissue for analysis, and all the patients give consent to participate in this sort of screening. One slide of their tumor is actually sent to MD Anderson, where a pathologist confirmed the diagnosis to make sure that they really had the disease we thought they had. And mutational analysis was performed often in the center where that patient was located, but in many cases, tumor samples were sent around to other institutions who had particular expertise in analyzing some of these targets. All of these data were fed into a database that we maintain. But importantly, I think, and maybe uniquely, all of this information came back to the treating doctor. And this was used to select therapy for the patient by the physician or to recommend clinical trials of agents that actually target the specific mutation of relevance. This is a snapshot of where we were in June of 2011. We had analyzed over 500 tumors for all of the mutations and the gene rearrangements. You can see we had a slightly larger collection of mutation profiling than of translocations, but a large overlapping set. And today, we actually have about double that now. This is what the mutations looked like and how they kind of broke down. And Dr. Shaw is going to speak a little bit about how these are characterized in different patient populations. The most common mutation is one called KRAS. This is a common mutation, one that we don't have a good way of targeting, but which we're actively exploring. The second most common is EGFR. We do have a targeted therapy that's quite effective, at least initially, for that subset of patients. The third subset was translocation of the chromosome called EML4-ALK, present in 9% of cases. And again, we have a drug that very specifically targets that Alice was one of the people that helped develop. And you can see there's a smattering of other mutations that were found that are very rare, but that when present, we think are important for these tumors. Overall, about 55% of the tumors had some detectable mutation. This was the patient characteristics when we did our initial analysis last June, looking at the first 500 and some patients. You can see that mutations are a little bit more likely to be present in women than in men overall, but really they can occur across age, across gender, and across smoking history. So despite some differences in the way these mutations track, we really encourage pretty broad screening for mutational profiling. I think looking forward, one of the most exciting things we now are beginning to do as a consortium, and this consortium is still actively working together even though the grant has run out, is that for many of these mutational subsets, we now have targeted therapy trials that are active in the group that are going forward. For KRAS, we have a study that involves erlotinib with another drug called RQL197. We think that may be active for KRAS mutant tumors based on very small numbers of patients, but some very exciting results that came forward and were initially presented by Joan Schiller. And as you can see, for all of the various subtypes, we now have a study that we think may be a path forward to at least develop new therapies for these different driver types of lung cancer. I think that's actually a very important outcome of the consortium, is that we have the ability to take even very rare mutations, say BRAF, which is only 2% of lung cancer, and begin to have studies that really focus on that rare but important patient population. And finally, just to wrap up, the goal of the Lung Cancer Mutation Consortium was to define the mutational frequency in these lung cancers. And I think an important result of that is we were able to find a mutation that we can actually do something about, at least direct therapy toward, in over half the patients with lung adenocarcinoma. The other thing I think this really showed us as a group is that multi-center coordination in this sort of large-scale way was feasible and actually quite effective. One outcome of this was improved comprehensive screening at individual cancer centers and, of course, facilitating target-directed therapy for these rare mutational subsets. I think this represents a model for the lung cancer community and other tumor types as well.
Great. One question I did want to ask as well, we already covered the issue of the different testing. What do those particular tests mean? But people will hear about CLIA certification. Can someone speak to, maybe Alice, if you can speak to CLIA certification, what that represents, and how important is that when people are getting testing done for any of the things we're talking about?
Yeah, my understanding is that CLIA certification is very important. It's a standardized way that labs are basically evaluated and judged to be capable and technically competent to do this type of testing for our patients. And ideally for all the genetic testing that we do, especially with regards to patients then enrolling on clinical trials, we would like to have that testing done in a CLIA certified lab. So that's why it was very important in the LCMC to have that testing done in these types of labs.
Although being a community oncologist, it's worth pointing out that a CLIA lab does not have to be a university-based center. It could be a community-based lab as well.
Absolutely. And Dr. Shaw and I were just discussing about the reliability of the tests, a case where initial screening at Johns Hopkins of a patient suggested that he had a translocation that involved the ALK gene. When that was tested in a central lab, that actually didn't confirm. Nonetheless, that patient had a great response to the drug that targets that mutation. So we think the reliability of some of these tests is not 100%.
And that's really a concern. We are actually seeing some reports, especially with the secondary mutations like T790M, of big differences between one lab's way of doing it and another, and these potentially have big clinical implications. So it's not just an issue of I got tested, but where and how you got tested. That could make a big difference. And my sense is that we're really only getting our own idea right now of how complex things really are.
I think the only thing we could perhaps learn from is the experience in breast cancer with HER2 testing, where in the beginning the community-based testing really was not very accurate. But in time and with a lot of education on the part of pathologists, that's gotten to be very, very excellent at all levels. So I think a lot of these things, as new markers are discovered and new testing is done, it just has to be done carefully where we first are sure we know how to do it before that gets translated to other sites so that patients, when they get tested, can believe the results.
Right.
I'm going to briefly speak about validation in my talk today.
Good. Because one of the big issues is, as this broadens from being something available at just a couple of leading centers to broad applicability in a wide community, how do we remain confident that the results are the same across a network? So one of the things that's important about the Lung Cancer Mutation Consortium was adenocarcinomas only. Mm-hmm. And obviously that's not everyone with lung cancer. One issue is why limit to adenocarcinomas, and then what are the factors that would make a difference between a cancer that is more likely to have a driver mutation and one that doesn't, and that doesn't lend itself as easily to the Lung Cancer Mutation Consortium approach?
I think the reason that we initially focused on adenocarcinoma was that many of these mutations seem to be more prevalent in that subtype. There was a lot of excitement when this was launched in 2009 about mutations that were occurring in patients with adenocarcinoma. So that's really where the focus was. In 2011, 2012, we're beginning to look at squamous cell carcinomas much more carefully and looking at mutations that may be important for those as well. This is the first phase of what may be a new approach.
Yes, and I think that adenocarcinoma is likely to be a simpler disease than squamous cell carcinoma, because squamous cell carcinoma is very strongly related to the amount of smoking that an individual has done in their lifetime, and therefore the genetic damage is probably much more extensive. Whereas many of the adenocarcinomas occur in non-smokers, the pathway to carcinogenesis would appear to be more simple. So I think that is another reason why you started with the adenocarcinomas. It's probably the easier of the two models to deal with.
And there's data supporting that even within the adenocarcinoma subset, patients with a heavier smoking history do have genetically more complex tumors when they do whole genome profiling. So we know that there is a very strong correlation between these simpler tumors that may be driven by a single oncogene and smoking history.
So I guess one key question is this going to be something that's going to potentially be or likely be broadly applicable to a wider range of people, or is this largely going to be most useful for people who have a minimal smoking history and an adenocarcinoma? I don't know if there's an answer.
Yeah, I think that's the holy grail. How complex is cancer? Not just talking about lung cancer, but pancreatic cancer, et cetera. Are we going to be successful in finding single driving or addicted models, so to speak? I'm sure Alice will get into that. Or are these going to be such heterogeneous tumors that how can you inhibit multiple pathways at the same time or sequentially? Is it going to require drugs that do that, single drugs or multiple drugs, or those drugs interact? I mean, this is at one point the most exciting area of oncology and at the same time the most daunting area. It's kind of scary to think that these complex tumors may be too hard to stop every pathway.
But I think it's a road that we have to travel because these mutations have taught us a lot about the biology of lung cancer. And as we go further along the road and discover more of these mutations and how they interact in more complex ways, we'll know more about the disease and we'll be able to treat a larger segment of the population.
We'll continue in the next podcast with Dr. Shaw speaking on the experience from Massachusetts General Hospital, profiling a broad range of their patients with advanced non-small cell lung cancer. Thanks for listening. If you like and learn from our GRACEcast, you can subscribe on iTunes by just searching for the term Cancer Grace, find podcasts in the subject you want, pick a format of audio or video, and then just click subscribe. It's that easy. And for those of you who don't want to miss any of our programs, there's even a feed for all subjects. You can also find us on YouTube at Grace for Cancer Info, and that's the number 4 in one word, Grace for Cancer Info. Finally, if you haven't been there yet, please check out our Grace website at www.cancergrace.org. And don't forget that donate button in the upper right. Our content, which helps tens of thousands of cancer patients around the world every month, is made possible by your support. Thanks again.